Okay, so the organelle involved in cellular respiration is the mitochondria. The main components of the mitochondria include the inner membrane, the intermembrane space, the outer membrane, mtDNA, the cristae, 70S ribosomes, and the matrix. So there are three main parts of cellular respiration. The first is glycolysis, which happens in the cytoplasm. The second is the Krebs cycle, which happens in the matrix. And the third is the electron transport chain in chemiosmosis, which happens in intermembrane space.
Okay, so basically cellular respiration is the controlled release of energy from organic compounds in cells to form ATP. During cellular respiration, energy flows from glucose to NADH to electron transport chain to proton and motive force to ATP. 4 ATP are produced directly by substrate level phosphorylation during glycolysis and Krebs cycle, with a 2 net ATP gain in each. Then, finally, 34 ATP are produced by oxidative phosphorylation.
A process called glycolysis, which is the breakdown of a 6-carbon molecule called glucose, which is a common sugar that we eat in our food and is commonly ingested in our body. Glucose is also is a common energy source for us. So, it's broken down through these 2 ATP molecules, which is adenosine triphosphate. The 6-carbon molecule is then broken down into 2 3-carbon molecules called pyruvate. Also, through this process, 2 ATP and 1 NADH is formed per 3-carbon molecule that is formed. Because of this, because there's 2 3-carbon molecules that are formed, there's a net gain of 2 ATP. This pyruvate is then used in the Krebs cycle, and the rest of cellular respiration is a key factor. Along with this, however, glycolysis can be used for other 3-carbon molecules, which can be used in fermentation without oxygen, and is also an important part of maintaining our energy and creating more ATP for our body to use. Fermentation takes place after glycolysis when there's no oxygen present in the cell. After glucose is broken into 2 3-carbon pyruvate, either lactic acid or alcoholic fermentation takes place. Through lactic acid fermentation, the NADH molecule that is released through glycolysis is used to convert pyruvate into 3-carbon lactic acid, and this NADH is then becomes an NAD+, which is then reused again later in glycolysis in another form. Also, for each fermentation process, 2 NADHs are used to convert the products. Another type of fermentation is shown on the bottom, which is alcoholic fermentation. In this process, first, the 3-carbon pyruvate is converted into acetaldehyde through the release of a carbon dioxide molecule. Then the NADH then is used to convert this acetaldehyde into a 2-carbon ethanol molecule, which again the same process takes place where they use 2 NADHs to transform it, and 2 NAD pluses are then formed as a result.
So following the production of pyruvate in glycolysis, the 3-carbon compound enters into the mitochondrial matrix, releasing CO2 and NADH. It creates a 2-carbon compound called acetyl coenzyme A, which then combines with 4-carbon compound to create a 6-carbon. The 6-carbon then goes to become a 5-carbon with the release of CO2 and NADH. This happens again to become a 4-carbon, and then this 4-carbon compound reacts with an enzyme to create ATP, and then the 4-carbon again reacts with an enzyme to create FADH2 and then NADH, and then the 4-carbon is used again for continuing the Krebs cycle. It's really important for the production of NADH for later in chemiosmosis and the electron transport chain.
Oxidative phosphorylation consists of both electron transport chain and chemiosmosis. The electron transport chain is electron transport and coupling of protons, which are the hydrogen ions, to create a hydrogen ion gradient across the cell membrane. Chemiosmosis is the ATP synthesis powered by the flow of hydrogen ions back across the membrane. In electron transport chain, electron carriers alternate between reduced and oxidized states as they accept and donate electrons. Each component of the chain becomes reduced when it accepts electrons from its uphill neighbor Then it returns to its oxidized form as it passes electrons to its downhill neighbor, which is the more electronegative neighbor. During oxidative phosphorylation, chemiosmosis then couples electron transport to ATP synthesis. Energy stored in a form of hydrogen ion gradient across the membrane is then used to drive cellular work, which is ATP synthesis, through the ATP synthase, which is an enzyme that makes ATP from ADP and inorganic phosphate.
This is glycolysis, and this is a 6-carbon compound called glucose, and breaks down into 2 ATP. And this cellular respiration begins with a process called glycolysis, which is the breakdown of a 6-carbon molecule called glucose, which is a sugar molecule commonly found in our bodies.
Oxidative phosphorylation.
That'll be in. Okay, ready?